Imagine for a second that there's a massive, highly toxic, hazardous waste spill right in the middle of a major, densely populated city.
Right, an absolute nightmare scenario.
Yeah, exactly. And typically, the response to something like that is entirely reactive. You mobilize the hazmat teams, you spray heavy chemical neutralizers everywhere.
You block off the streets.
Right. You block off the streets and you spend billions of dollars trying to scoop up the mess before it seeps into the groundwater. It's just this exhausting, incredibly expensive battle against the symptoms of the spill.
It's pure damage control.
Completely. But what if, instead of sending in a hazmat crew in protective suits, you could just, plant a handful of seeds in the middle of that spill?
Okay, that's a completely different approach.
Right. And what if the biological architecture of the plants that grew from those seeds was specifically designed to drink up that toxic waste, use it as fertilizer, and basically breathe out completely harmless, clean air?
It completely flips the paradigm. You're talking about taking a devastating environmental threat and turning it into the literal fuel for the cleanup process.
Yeah.
It changes the framework entirely from a defensive posture of, how do we fight this hazard, to a biological integration of how do we naturally metabolize it.
Exactly. And that fundamental shift in thinking is the mission of today's deep dive. We are exploring a revolutionary concept in microbiology that could fundamentally change how we treat illness, how we protect our global food supply, and really how we understand the microscopic ecosystems living inside our own bodies.
It's a massive topic.
It really is.
Right.
We are looking at microbial detoxification. And what's brilliant about this idea is that it isn't about inventing some highly synthetic, complicated, expensive new pharmaceutical drug. It's about uncovering a hidden superpower in something you might already have sitting in your kitchen cabinet. We're talking about everyday probiotics.
Which is highly relevant because so many of you listening are probably already taking a daily probiotic.
Oh, for sure.
You might take a capsule with your morning coffee, or you eat a bowl of fermented yogurt, mostly with the vague goal of supporting general gut health, maintaining your microbiome, or, just aiding digestion.
We tend to think of them as these gentle, passive little helpers, just keeping our stomachs calm. But this deep dive is going to reveal how those same microorganisms might soon serve as an active, aggressive, internal cleanup crew. Okay, let's unpack this.
Let's do it.
Before we get into the biological mechanics of the solution, we have to understand the sheer scale of the problem we are trying to solve. We aren't just talking about fixing a minor stomachache.
No, not at all.
We are looking at a global crisis. So, to start us off, set the stakes for us. Why do we even need a radically new approach to detoxification?
Yeah.
Haven't we pretty much figured out pathogen control with modern medicine and modern agriculture?
You would certainly think so, given the advancements of the last century. But the reality is actually quite grim. We are facing a massive, largely invisible threat, and our current modern arsenal is, well, it's profoundly failing.
Wow.
Yeah. We are looking at two separate but equally devastating global crises. The first is mycotoxins.
Right, the molds.
Exactly.
Right.
These are highly toxic secondary metabolites produced by certain molds and fungi. And they aren't just rare contaminants found in damp basements. Mycotoxins are pervasive in the agricultural environment. They contaminate a staggering 25% of the world's global food crops.
Wait, 25%?
25%.
That is 1/4 of the entire global food supply laced with invisible toxins before it even hits a plate.
And because it's so widespread, the health outcomes are severe. When you ingest these mycotoxins, things like aflatoxins or ochratoxins, they don't just pass through you. They are actively hepatotoxic and immunosuppressive.
Meaning they attack the liver and the immune system.
Exactly. And to understand the how here, you have to look at what they do on a cellular level. When certain mycotoxins hit your liver enzymes accidentally convert them into highly reactive compounds called epoxides. These epoxides then bind directly to your DNA. They literally wedge themselves into the genetic code, which causes mutations. This directly drives massive rates of liver cancer.
So the body's own filtration system inadvertently weaponizes the toxin against itself.
Precisely. And in vulnerable regions, chronic exposure to mycotoxins leads to severe stunting in children.
Oh, wow.
Yeah, the toxins constantly inflame and damage the intestinal lining. It's a condition often called environmental enteric dysfunction, meaning the child's gut simply cannot absorb nutrients anymore, no matter how much food they eat. Their physical and cognitive growth is permanently arrested. So that is crisis number one.
And crisis number 2.
Antimicrobial resistance, or AMR. This is the phenomenon where bacteria evolve to completely resist the antibiotics we use to kill them. It is already a runaway train, currently killing 1.27 million people every single year.
To put that in perspective, that means antimicrobial resistance is already killing more people globally than malaria or HIV. It's a terrifying statistic.
It really is, and the trajectory is even worse. By the year 2050, epidemiological projections indicate that antimicrobial resistance could kill up to 10 million people annually.
10 million a year.
Yes, and the mechanism behind AMR is simple evolutionary pressure. When we use traditional antibiotics, we are essentially carpet bombing the microbiome. We wipe out all the susceptible bacteria, good and bad.
Right.
But if even a fraction of a percent of the pathogenic bacteria have a random genetic mutation that lets them survive the antibiotic, they now have zero competition for resources. They multiply rapidly, and worse, they can literally trade those resistance genes with other bacteria using tiny loops of DNA called plasmids.
So we have cancer-causing mycotoxins permanently woven into our food supply and unstoppable superbugs trading genetic armor in our hospitals.
It's a perfect storm.
And when you look at how we currently try to solve these massive problems, I mean, our current solutions are essentially a dead end. Right now, to deal with mycotoxins in agriculture, we rely on chemical binders added to animal feed or incredibly expensive filtration processes.
Which a lot of places simply can't afford.
Exactly. And for AMR, we just desperately try to synthesize newer, harsher, and more expensive antibiotics. The shared flaw among all these methods is that they are purely reactive. They only treat the symptoms after the damage is done. They are incredibly costly, making them completely unsustainable for the global population.
And they systematically fail to reach the billions of people in developing nations who actually need them the most. What's fascinating here is how these two massive threats, mycotoxins and antimicrobial resistance, are treated by the modern medical and agricultural establishments as entirely separate, unbreakable problems.
Like they're two different wars.
Right. We are trying to solve both of them using artificial, highly synthetic chemical means that don't actually integrate with the biological systems we are supposedly trying to save. We're fighting millions of years of microbial evolution with static chemicals, and evolution is winning.
It's totally ineffective. Honestly, using chemical binders and broad-spectrum antibiotics right now is like frantically bailing water out of a sinking boat with a leaky bucket.
Yeah, that's a good way to put it.
Or really, it's like trying to put out a raging forest fire by swatting at the smoke instead of just taking away the fuel. We are just exhausting our resources while the fire spreads faster and faster.
The bucket is leaking, the firefighters are exhausted, and the fuel keeps piling up. Which brings us to the pivotal question.
What if we just take away the fuel?
Exactly. Because the current methods are failing so spectacularly, there is a massive push to pivot entirely. The idea is to repurpose something that is already cheap to produce, globally scalable, and critically already proven to be completely safe for human and animal consumption.
Which brings us back to the probiotics. But we really need to clarify what probiotics currently do versus what this new paradigm proposes they could do. Because traditionally, as anyone who follows health and wellness knows, we view probiotics as these live microorganisms that confer general health benefits.
Right, the good guys.
Exactly. They are primarily known for promoting gut homeostasis. They provide stress tolerance, they have some basic hydrolase activity to help break down food, and they offer passive antimicrobial effects. They are the resident good guys.
Yes, the established scientific consensus views them primarily as maintainers of balance. They occupy the ecological niches in your gut. But the breakthrough question, the one that changes the whole game, is this. Can probiotics actively degrade complex environmental toxins instead of just passively competing with pathogens for space?
Wait, let me push back on that for a second. I really want to make sure the mechanics of this make sense. Normally, we think of probiotics as taking up parking spaces in the gut, right?
Yes.
They park their cars, so the bad guys have nowhere to park, and therefore the pathogens can't establish a colony.
Huh.
But if you're suggesting they're actually walking around disarming the bad guys by eating their highly toxic weapons,
Yeah.
how do they survive?
It's a fair question.
I mean, if these mycotoxins are powerful enough to shred human DNA and destroy liver cells, why don't they just melt the probiotic bacteria?
Yeah.
How does a single-celled organism eat poison and live?
That is exactly the right question to ask, and it gets right to the heart of the biochemistry. The reason the probiotic doesn't melt is because it doesn't process the toxin the way our liver does.
Okay, so it has a different pathway.
Remember how the human liver accidentally converts the mycotoxin into a DNA-damaging epoxide?
Yeah.
Well, the probiotic bacteria don't make that mistake. Instead, they have evolved highly specific enzymatic pathways. They produce targeted enzymes like lactanases or amidases that act like microscopic molecular scissors.
Scissors. I like that visual.
Right, and when they encounter the mycotoxin, these enzymes precisely cleave the specific chemical bonds that make the molecule toxic. They break the toxic rings apart.
So they dismantle the bomb before it can go off.
Exactly. And once those toxic bonds are broken, what's left is just basic carbon and nitrogen. The bacteria then use those broken down elements as a carbon source. It literally becomes food for them.
That is wild.
It is. We are moving from viewing probiotics as passive space occupiers, just holding down the fort, to viewing them as active agents of targeted biochemical degradation. This represents the very first probiotic platform with a built-in detox function. It's not just about restoring harmony, it is a fundamentally different way to wage microbial warfare.
Here's where it gets really interesting. Because we aren't just talking in abstract theoretical biochemistry. We have specific, real-world examples of bacterial strains that are already doing this right now. The prime candidate often discussed in the sources is Bacillus subtilis.
Bacillus subtilis is the perfect example to prove viability. It is already commercialized, highly adapted, completely safe for consumption, and has a proven track record as a health-benefiting supplement, particularly with major success stories in regions like India.
So it's not some fragile lab experiment.
No, not at all. This isn't some genetically modified organism that was stitched together in a sterile lab yesterday. It's a robust, naturally occurring microorganism.
But to really stress test this idea, we have to look at the environment it's operating in. It's one thing for Bacillus subtilis to secrete organic acids and enzymes in a perfect, temperature-controlled petri dish. But the human gut, or a cow's rumen, or agricultural soil, is incredibly messy.
Oh, it's a war zone.
A total war zone of pH changes, digestive acids, and competing microbes. The big question is whether this enzymatic degradation can occur robustly in complex microbial environments. Can it establish itself despite the interference of millions of other unwanted organisms?
Which is the make-or-break hurdle for any biological intervention. Ecological compatibility and extreme resilience are mandatory. If the probiotic gets destroyed by stomach acid or gets bullied out of existence by the resident microbiome, the detox function never happens.
Makes sense.
And that is why Bacillus subtilis is so special. It is a spore-forming bacteria.
Meaning it can essentially build a bunker around itself.
Exactly. When it faces a harsh environment, like the highly acidic human stomach, it forms a tough, protective endospore. It goes dormant, survives the acid bath that would kill most other probiotics, and only germinates and wakes up once it reaches the friendly, nutrient-rich environment of the intestines.
And to guarantee that survival and integration, we have to talk about prebiotics. Now, you usually hear prebiotics described simply as indigestible fibers, things like inulin or oligosaccharides. But in this context, they serve a much more strategic purpose.
They do.
They're a dedicated home and fuel source for the probiotics. If the probiotic is the specialized worker sent in to do the detoxifying, the prebiotic is the logistical support system. It's like sending a specialist into a highly hostile, contaminated zone. But you send them in with a fully packed lunch and a cozy, reinforced tent.
I love that.
You are ensuring they have the exact metabolic energy and the physical shelter they need to establish a foothold in the messy ecosystem of the host's body and actually perform the enzymatic cleavage of those toxins.
The packed lunch and the cozy tent is a brilliant way to visualize it. It highlights that this process isn't happening in a vacuum. It requires deep integration into the host's existing biology. And that integration is what makes a biological intervention so much more powerful than a blunt chemical binder.
Right.
Because when you introduce these specific probiotics and their organic acids into that complex ecosystem, the downstream effects ripple outward in ways that transform the entire environment.
Which leads us to what is, without a doubt, the most mind-bending revelation of this entire concept. We've talked about how the probiotics survive, how they eat the toxins, how they degrade the poison into harmless carbon. But what does this mean for the harmful pathogens themselves?
The ones actually making the toxins.
Exactly, the actual bad bacteria that are producing these toxins in the gut to begin with. Because it's not just about destruction, it's about ecological reprogramming.
This is the crux of the entire paradigm shift. We have to stop thinking of this as a drug that kills things. It is a fundamental alteration of an ecological relationship. Think about the mechanics of an infection. Pathogens don't just produce toxins for fun.
They have a strategy.
They do. They use a communication system called quorum sensing. They release signaling molecules to count how many of their friends are nearby. Once they reach a certain density, they all collectively release their toxins to overwhelm the host's immune system. That's how they conquer the territory.
It's a coordinated microscopic ambush.
Exactly. But now introduce our actively detoxifying probiotics, like Bacillus subtilis. They are biosynthesizing organic acids and enzymes. They aren't just breaking down the mycotoxins, they are degrading the pathogen's signaling molecules too. They are literally eating the enemy's communication network and their weapons, using them as metabolic fuel.
So they are blinding the pathogen and disarming it simultaneously.
Yes. And here's where the reprogramming happens. Because the pathogen's weapons are instantly neutralized and turned into food for the probiotic, the pathogen loses its competitive advantage. The evolutionary pressure completely shifts.
Wow.
The pathogen realizes, in a biochemical sense, that expending energy to create toxins is useless. So it downregulates its virulence genes, it stops attacking, it is forced to adapt to the new environment, potentially converting from a harmful, aggressive bacteria into a harmless commensal. It just becomes part of the normal, quiet background flora. You make friends with the pathogen, you turn enemies into allies.
That is unbelievable.
If we connect this to the bigger picture, you can see how this one ecological mechanism tackles both the cancer-causing mycotoxin crisis and the antimicrobial resistance crisis at their absolute root source. You are enzymatically eliminating the environmental mycotoxins before they can reach the liver and mutate your DNA.
Right.
And simultaneously, you are neutralizing deadly pathogens without ever firing a single shot of traditional antibiotics. You never create the evolutionary vacuum that breeds superbugs.
So instead of dropping a chemical bomb on the gut microbiome with broad-spectrum antibiotics, hoping we kill the bad guys before they mutate, we are sending in highly specialized microbial diplomats.
Diplomats, exactly.
They literally eat the enemy's poison for breakfast, dismantle their communication lines, and alter the environment so thoroughly that the pathogens just give up and join the neighborhood watch.
That is precisely it. It is diplomacy and ecological engineering on a microscopic scale. You aren't trying to eradicate the pathogen, which we know just breeds resistance. You are ecologically reprogramming its behavior. You take away its weapon, you consume its toxins, and you integrate it safely back into the normal ecosystem. It is a profound shift from a scorched earth war mentality to a sustainable ecological mentality in medicine.
It's just staggering to think about the downstream implications of this. We are looking at a biological solution that isn't just a neat biochemical curiosity confined to a university lab. This is a highly scalable, eco-friendly, completely biodegradable solution to some of the world's most pressing and fatal crises.
The scope of who benefits is practically everyone. This would be absolutely life-changing for farmers and consumers in mycotoxin hotspot regions. If you deploy this in agricultural sectors in India, sub-Saharan Africa, and parts of Asia, where that 25% crop contamination hits the absolute hardest, you directly intervene in the devastating rates of childhood stunting and liver cancer. You protect the food supply at the source.
And it benefits patients worldwide who are terrified of antimicrobial resistance. We are talking about fundamentally altering the trajectory of that projected 10 million annual deaths by 2050.
It could save millions of lives.
And on a consumer level, it points toward a future line of products that literally turn everyday food into preventative medicine. Imagine eating your daily yogurt but knowing you are arming your body with an active, heavily shielded detox crew that chemically dismantles carcinogens.
It's a completely different way to look at nutrition.
So what does this all mean? The core vision here is incredibly elegant. A daily probiotic that doesn't just passively sit there and help your gut digest fiber, but actively seeks out and eats the poison before it can harm you. It is entirely preventive, it works in lockstep with the body's natural ecosystem, and it tackles complex diseases at their chemical root.
This raises an important question, one that pushes this concept even beyond the bounds of human gastroenterology. We focused heavily on the human and animal gut, but the underlying mechanics here rely on environmental microbiology. These biodegradable pathways, these enzymatic scissors, they explore how these probiotics interact with the broader environment.
Right, because mycotoxins don't originate in the human stomach. They originate in the dirt. They are fungi growing on the agricultural crops out in the fields.
Exactly. The contamination starts in the soil.
So here is a final thought for you, the listener, to mull over long after we wrap up today's deep dive. If we know that these specific resilient probiotic strains can act as highly efficient eco-friendly detoxifiers. And if we know they can ecologically reprogram aggressive pathogens and survive incredibly complex, messy environments like the human digestive tract, could we eventually see a world where we deploy these exact same microscopic peacekeepers directly into contaminated agricultural soils?
That's a fascinating possibility.
Could we formulate a robust probiotic for the earth itself, actively consuming and neutralizing the environmental toxins in the dirt before they even have a chance to touch the roots of the plants we grow?
It completely reframes our entire relationship with the microbial world. We stop fighting it and start collaborating with it.
It really does. Because in the end, you don't always need to send in a massive hazmat crew with chemical sprayers to clean up a toxic spill. Sometimes you just need to plant the exact right seed and let the biology eat the poison.